The small intestine plays a key role in the digestion and absorption of nutrients. 90% of nutrient absorption occurs in the small intestine and the rest in the large intestine. The small intestine is about 20 feet long and has 1 to 1.5 inches in diameter. It's made up of 3 segments: the duodenum, the jejunum, and the ileum. The duodenum is close to the stomach. It's a C-shaped curved portion of the small intestine that wraps around the pancreas. It receives chyme from the stomach, plus liver and pancreatic secretions, and it's mostly retroperitoneal or behind the peritoneum. The spot where the small intestine re-enters the peritoneum is the jejunum. It's pictured here in purple. Most chemical digestion and absorption occurs in the jejunum. The ileum, the pink portion of the small intestine, is the longest portion, and it ends at the ileocecal valve that controls the flow into the large intestine. Note the permanent circular folds that create transverse ridges on the cross-section here. Those circular folds are just one of the 3 levels of increased surface area in the small intestines. There's also villi, which are finger-like projections of mucosa covered with simple columnar epithelium, and the microvilli, seen here, which are modified apical surface of the columnar cells, and it's labeled in this picture as the brush border. Each of the villi has an intestinal gland that secretes mucus and intestinal juice. You can see these in the bottom left picture. Capillary beds are also present to transport gases and absorb nutrients to the hepatic portal system. And you'll notice that there's a lymphatic capillary present to transport material that can't enter the bloodstream, like fatty acids. And these are packaged into chylomicrons. So after chyme has entered the duodenum or duodenum, weak peristaltic contractions move it slowly towards the jejunum, and these contractions are local reflexes, not under central nervous system control. More elaborate reflexes coordinate activities along the entire length of the small intestine. Distension of the stomach initiates the gastroenteric reflex, which accelerates glandular secretions and peristaltic activity and empties the duodenum. The gastroileal reflex is a response to circulating gastrin, which relaxes the ileocecal valve, and then material can move from the ileum into the large intestine. Roughly 1.8 liters of watery intestinal juice enters the intestinal lumen each day. Much of this fluid arrives by osmosis from the mucosa, and the rest is secreted by intestinal glands. Intestinal juice moistens the internal contents. It buffers acids, especially in the duodenum. This is important. And it keeps both the digestive enzymes and the products of digestion in solution. Some intestinal hormones include gastrin, which is made in response to a high-protein chyme and stimulates the production of acids and enzymes. Secretin is made in response to low pH chyme, and it increases buffers and bile secretions. Choleocytokinin, or CCK, is made in response to high-fat chyme. In the pancreas, it increases enzyme production, and in the gallbladder, it causes the ejection of bile. Gastric inhibitory peptide, or GIP, is made in response to high fat and high glucose chyme. In the stomach, food becomes saturated with gastric juices and exposed to the digestive effects of a strong acid, hydrochloric acid, and proteolytic enzymes like pepsin. Most of the important digestion processes are completed, however, in the small intestine, where the final products of digestion, like simple sugars, fatty acids, amino acids, are absorbed, along with most of the water content. The small intestine produces only a few of the enzymes needed to break down the complex materials found in the diet. Most of these enzymes and buffers are contributed by the liver and the pancreas. So let's take a closer look at the pancreas. It lies behind the stomach and extends from the duodenum towards the spleen. It is retroperitoneal. Some of the major tissues and functions in the pancreas include the pancreatic islets, which are endocrine cells I told you about in the endocrine chapter. These secrete insulin and glucagon. You'll also see the pancreatic acinar cells, which are the exocrine portion of the pancreas. These produce digestive enzymes and buffers, and these cells are organized into pouches called pancreatic acini. There are duct networks that converge into the pancreatic duct, which then empties into the duodenum. There are 4 classes of pancreatic enzymes: carbohydrases, lipases, nucleases, and proteases. Each day the pancreas secretes about 1 quart of pancreatic juice. Secretions are controlled primarily by hormones from the duodenum or the duodenum. When acidic chyme arrives in the duodenum, secretin is first released, and this triggers the pancreas to secrete a watery alkaline fluid containing sodium bicarbonate with a pH between 7.5 and 8.8. Another duodenal hormone is the CCK that stimulates the production and secretion of pancreatic amylase to break down carbohydrates, lipase to break down fats, and proteases like trypsin, chymotrypsin, and carboxypeptidase. Next, the liver. The liver is found in the right and epigastric abdominopelvic regions. It's encapsulated and covered by visceral peritoneum. It has 4 lobes, a large left lobe and a large right lobe. Then we have a smaller caudate and quadrate lobes. On the anterior portion, we have a falciform ligament, which is the division between the left and right lobes. The thickened posterior part of the falciform ligament forms the round ligament, which, if you recall, is a remnant of the fetal umbilical cord. Liver lobes are divided into about 100,000 liver lobules. These are the functional units of the liver. They contain hepatocytes, which are covered in microvilli, arranged in plates like spokes on a wheel. We have a portal triad found at each of the 6 corners of a lobule and contains a branch of the hepatic portal vein, a branch of the hepatic artery, and also the bile duct pictured in green here. Sinusoids are specialized capillaries that empty into the central vein. They contain Kupffer cells for phagocytosis. So blood comes from the hepatic artery and the hepatic portal vein, and as it flows through the sinusoids, hepatocytes absorb nutrient molecules and secrete plasma proteins. Blood leaves the sinusoid into the central canal and merges to form the hepatic veins, which drain into the inferior vena cava. Those hepatocytes also are responsible for secreting bile into bile canaliculi between adjacent liver cells, and they empty into the bile duct in a triad. Bile then flows into the common hepatic duct and into the common bile duct, and from there into the duodenum. If hepatopancreatic sphincter is open, it will flow in, or if it's closed, it'll back up into the cystic duct and be stored in the gallbladder. The gallbladder is a hollow pear-shaped organ tucked up under the liver, and it stores and concentrates bile. CCK, if you'll recall, triggers contractions of the gallbladder and relaxation of the hepatopancreatic sphincter, so concentrated bile can flow into the duodenum. And concentration of the bile is a very efficient means of storage. As I mentioned, hepatocytes extract nutrients and toxins from the blood as it flows past them in the sinusoids, as well as monitor and adjust the circulating levels of organic nutrients. Excesses are removed and stored, and deficiencies are corrected by mobilizing stored reserves or synthesizing the necessary compounds. The liver is the largest blood reserve in the body, and as blood passes through the liver, phagocytes remove aged or damaged red blood cells, any debris, or pathogens. Hepatocytes synthesize plasma proteins that keep the osmotic concentration of the blood consistent, like those involved in clotting and the complement cascade. Hepatocytes synthesize bile and they excrete it into the duodenum. Bile consists mostly of water and ions to dilute and buffer acids in the chyme. It also contains bilirubin from the breakdown of hemoglobin, cholesterol and lipids called bile salts, and these are used to emulsify large fat globules into smaller ones. And the idea here is that emulsification will increase the surface area of the lipid for enzymatic action. So we've reached a checkpoint. I'm going to take a break from the recording and answer these questions. The large intestine begins at the ileocecal valve and ends at the anus. It is a horseshoe-shaped organ that frames the small intestine, and it functions to reabsorb water, compact chyme into feces, and absorb vitamins freed by bacterial action. It's also responsible for the storage of feces prior to defecation. The cecum is an expanded pouch that chyme enters through the ileocecal valve, and the cecum functions to initiate compaction. The appendix on the postremedial surface contains lymphoid nodules that may function for the immune system. The colon has a large diameter, it lacks villi, and has abundant mucus cells. Externally, it has the haustra, which are pouches that distend and elongate. The tinea coli are 3 longitudinal bands of smooth muscle, and the large intestine is subdivided into 3 segments: the ascending colon, transverse colon, descending colon, and the sigmoid colon. The rectum is an expandable organ for storing feces. The anal canal is the last portion, and it contains longitudinal anal columns. The anus is the exit of the anal canal, and it has both an internal anal sphincter and an external anal sphincter. The internal anal sphincter is made of smooth muscle of muscularis externa, and it is under the autonomic nervous system control, whereas the external anal sphincter is voluntary skeletal muscle. As I mentioned, the large intestine functions to reabsorb water. It reabsorbs about 1.5 liters of chyme that enter the cecum each day. Of the 1.5 liters of chyme that enters the cecum each day, about 1.3 liters are reabsorbed. The large intestine also reabsorbs bile salts, which enter the hepatic portal circulation to return to the liver. Bacteria in the colon make 3 key vitamins: vitamin K, which is needed for the production of clotting factors. Bacteria make biotin, which is essential for glucose metabolism. And bacteria make vitamin B5, which is required for the synthesis of neurotransmitters and steroid hormones. Additionally, bacteria convert bilirubin into forms that are eliminated in the urine, giving the urine the yellow color, and non-absorbable forms that remain in the feces, giving it a brown color. Bacterial action also breaks down peptides into ammonia, nitrogenous compounds, and hydrogen sulfide. The ammonia and other toxins are absorbed, and the liver converts them into harmless forms, which is then eliminated by the kidneys. Indigestible carbohydrates provide a nutrient source for bacteria. However, it also forms flatulence or gas. The gastroileo and gastroenteric reflexes move material into the cecum while you eat, And from the cecum to the transverse colon, movement is very slow, allowing time for water absorption. Powerful peristaltic contractions called mass movements force the fecal matter through the rest of the large intestine. Distension of the rectal wall triggers defecation reflex, which involves two feedback loops. In the short loop, stretch receptors in the rectal wall stimulate local peristalsis, which moves the feces towards the anus and distends the rectum. In the long loop, it stimulates parasympathetic reflex, which triggers involuntary relaxation of the internal sphincter, but conscious relaxation of the external sphincter is still required for passage of the feces. A typical meal contains a mixture of carbohydrates, proteins, lipids, water, electrolytes, and vitamins. The digestive system handles each component differently. Large organic molecules are typically insoluble and must be broken down. Small organic molecules that can be absorbed are then released into the bloodstream. Once absorbed by cells, they're used to produce ATP, or to synthesize complex carbohydrates, proteins, and lipids for use in the cell. Water, electrolytes, and vitamins can be absorbed without preliminary processing, but may require special transport proteins or mechanisms. I like this figure because it shows the carbohydrates, lipids, and proteins and the chemical events in their digestion. You'll see that amylase breaks down complex carbohydrates into disaccharides and trisaccharides. And enzymes at the epithelial surface break these molecules into monosaccharides that are absorbed by the intestinal epithelium through facilitated diffusion or co-transport. Triglycerides are emulsified into large lipid droplets. The resulting fatty acids and other lipids interact with bile salts to form micelles, from which they diffuse across the intestinal epithelium. And these are packaged into chylomicrons, which are released into interstitial fluid and transported to the venous system by the lymphatics. Protein digestion involves low pH and the enzyme pepsin in the stomach, and various pancreatic proteases in the small intestine. Pepsidases liberate amino acids that are absorbed by the intestinal epithelium and then released into interstitial fluids. Intestinal epithelial cells absorb dissolved nutrients and ions. Sodium and chlorine are key in promoting water absorption, and wherever salt is, water will follow through osmosis. Other ions include potassium, magnesium, iodine, carbonic acid, iron, calcium. Calcium absorption, remember, is under control of the parathyroid hormone and calcitriol. Fat-soluble vitamins like A, D, E, and K are absorbed in micelles along with lipids. Vitamin K also is produced in the colon by bacteria. Water-soluble vitamins like B vitamins and C vitamins are easily absorbed by the digestive epithelium, except for B12, which requires that intrinsic factor. Bacteria in the gut are also a source of water-soluble vitamins. And that brings us to our final checkpoint and the end of this recording and this chapter.